Hello, everyone. Thank you for joining us today. This webinar will begin in just about a minute. We will wait a few more moments to allow attendees to join. I would like to welcome you to today's Zeiss virtual workshop. We will learn how to use artificial intelligence to drive efficiency in your microscopy setup. My name is Colleen Manning, and I am the Product Marketing Manager for Wide Field and Automation. I'm here with Dr. Samantha Four, the Product Marketing Manager for Laser Scanning Microscopy. We will be your hosts today. We also have joining us Dr. Frank Vogler and Gert Sontag, live from Germany. Gert is an application specialist for Zeiss and an expert in 3D imaging methods. He will provide an overview of the new AI sample finder and an outline of what we will be covering today. Operating the microscope is Dr. Frank Vogler. He is currently the regional sales manager in Germany, but previously was an application specialist like Gert. Frank will be running through some of the exciting application examples to highlight new hardware and software features in Zen 3.4. The workshop introduction and demonstration should take approximately 45 minutes, after which we will have about 10 to 15 minutes to answer some of your questions. Frank will first show you AI sample finder tiling on fixed brain sections, followed by an example of cell culture in 96 well plates. Please use the questions function in your dashboard. We will collect all of them and answer them at the end of this webinar. Any questions we do not get a chance to respond to, we will connect with you directly. In case you have any technical issues, please use the chat function in your GoToWebinar dashboard, and we will try our best to help you. We will be recording today's webinar, and it will be made available on the website after the live demonstration. I think we are just about ready to start. Without further delay, I will hand this over to our first speaker today, Gert.
Thank you, Colleen. Thank you, Samantha. Hello, everybody. Let's start with some basic understanding, and let's ask, why do we think we need a thing called sample finder based on artificial intelligence, and how does it may help to add efficiency to your daily routine? Being with Zeiss as a salesperson and application specialist for quite a long time, I like such a warm welcome. Somebody being happy with the latest Zeiss microscope. 8 hours sounds like a long day, but I'm quite sure at least 1 hour has been spent on picking the next position, picking the next sample, traveling back and forth all the time. With AI Sample Finder, we will learn that this is not only a simple tool to give you an overview or a pre-scan, but a lot more. It starts with assisted sample loading, carrier recognition. It gives you an overview image. It will detect the cover slip if there is one, and starts segmentation and gives you a coarse focus orientation. A lot of what AI Sample Finder does has to do with saving time, playing safe, and streamlining your workflow. It starts again with the automatic identification of the sample carrier type. It makes samples visible, clearly visible, even if they are translucent. You will never miss a sample on your slide again, or may do one of those samples twice without knowing. I've met a lot of people switching permanently back and forth between locate mode and acquisition mode in ZenBlue, and they just want to focus at the next zebrafish and find the right spot and save that position. So maybe this is one thing of the past. It's about time again, and it's a lot more. I might point out here, assisted loading. No more crashes between objectives and stage, or objective and your precious samples. It will ID the wells and cover slips. There's a unique composite dark field contrast, which allows to get images very gentle, even with no staining in your sample. Artifacts like fingerprints will get detected and excluded from acquisition. And last but not least, sample segmentation and identification preparing of your tile setup for the ongoing experiment is done at the very end of it. So why artificial intelligence? We have created neural networks by feeding lots of different samples, carriers, formats into that system, stained samples as well as unstained ones. And if you like, AI Sample Finder brings somehow intelligence into your microscope. So it's not just about sample finding. And that's the way it looks like if you enter such a setup. It consists of an inverted microscope, latest release, and you will see a multi-LED array at the back of your condenser. That's the one we take for giving us these unique dark-filled contrast images to segment the carrier as well as the sample. Hidden from view is a mirror and an overview camera in the objective turret. And if we look at a schematic like this, it becomes maybe more clearly than before. So we do have this LED array, we do have a sample. In the objective turret, we'll see this mirror. And at the back of your objective revolver, you have this sample camera or overview camera. And another set of LEDs in the IR range will help to focus at the sample. For this, we don't use the mirror anymore, but a low mag objective. And even with samples which does give close to no contrast, we'll be able to bring this in focus. Okay, the workflow in a nutshell, because it will be so quick once Frank is starting to operate that system. I'll run you through what the sample finder does step by step with some slides. Starting with a slide like this, which clearly shows some cross sections. I might have used the loading assistant with one of the clicks at the very top of this wizard, and then it starts automatically doing this overview image, getting the sample very close in focus, starting carrier detection, sample detection, and some 30 seconds later, I'm already at this point. And from this point on, this wizard hands over what he has found into the acquisition tab. And I assume we already have set up an acquisition experiment template, which allows to do something like transmitted light tiling, for instance. And we are ready to proceed and outperform this tiling experiment. Another minute later, mission accomplished, and we get the images we wanted to have. So there's no further interaction needed. You'll see this in the live session. And yeah, so for a slide with some cross sections, if you prefer to use jumbo slides, no problem. Only AI Sample Finder does not give you the positions of some cell clusters. It's too small, we won't see. But it helps to do the calibration. It does kind of a sample carrier recognition. It knows it's a 8-fold jumbo slide. It will outline each single chamber, and this helps to apply your precision pattern in the next step, and it gives you very safe mind once you are operating and navigating through this carrier. Same thing with a multi-well plate. Same thing here, we do the calibration, carrier recognition, and the system does know where each single well is positioned. And not only this, we get kind of a naming, the proper naming of all those carriers. All right, what do we need in terms of hardware and prerequisites to use AI Sample Finder? We definitely need the latest model of Axio Observer 7, the inverted stand, which with this special condenser and this mirror and this overview camera. We do need a scanning stage. We do need one of the low mag objectives, 2.5, 5x or 10x. One of the AxioCam models, latest releases, a monochrome one. This is what we need for focusing. And we definitely need software. It's then blue, 3.3 or higher, and a very powerful PC to allow all these algorithms based on artificial intelligence and make this very quick. You may ask yourself whether you can retrofit your already existing system. Yes, please talk to your service engineer or the salesperson in your area and find out what is needed to be added and to make this accessible on your system. A few samples before I hand over to show everything in live in action. So again, some slides with some cross sections. So if you look at this, I think it's no big deal to find them, but it's but applying the tiling pattern is another story. It will take time. It's very time consuming. So helps. Yeah. If you look at samples like this, everybody knows this neatly stacked embryos, one side by side. We can clearly identify them and take the locate mode again to travel, but much easier if you get them detected and you just need your screen to go to the next and next and next and apply the imaging settings to them. And last but not least, this is a slide with some translucent cross sections of a mouse brain. We don't see them by eye. This unique dark field contrast makes them available. And as we can see here, we do see the outline of the cover slip. We see all of them properly detected. And this is exactly the time where you can switch over into what you really want to do at the end, going with a higher magnification, applying some fluorescence on it, acquiring stacks and all this. All right. I would say now it's time for the live session, and I hand over to Frank. Thanks for listening so far.
So hello everybody. It's my pleasure to do the live session today. My name is Frank, and thanks Colleen, Sam and Gert for introducing here. So I'm sitting here next to an Axio Observer wide field system. Let me quickly introduce the system to you. So that's the microscope, the star for today. And as you see, it's being equipped with an AI sample finder. And as introduced, I've brought two samples today. The first one will be a slide with an array of mouse brain sections, and the second one will be a 96 well plate with some cell culture in it, which is fixed and stained. All right, so let's get started. Let me show my screen here and switch over to the microscope. Okay. All right, so you can see just a second. All right, so you can see the microscope interface here. That's our ZenBlue software. And let me just start the first experiment for today. So the sample finder is basically a single button click away. So you can find it in your acquisition tab. And with a single click, just get started into the AI sample finder dialog. To run the AI sample finder, it's basically just another single click to start the whole process of the different illumination techniques Gert introduced to you. Plus, there's an additional feature we introduced, and that's loading positions. There are additional loading positions which are here indicated and which will move the stage to the lower right or lower left position so that you can easily put your sample on the microscope without having to flip back the condenser arm. All right, so let's start the AI sample finder. And what you can see now is that there are several images being acquired with different kind of illumination, with the reflected light illumination, plus with a composite dark field contrast image, which is being based on the 128 LEDs in the back of the condenser. And this is now running all fully automatically through it, and the neural nets that are integrated in the AI sample finder will do several jobs. First of all, they will identify the carrier type. So in this case, it will automatically recognize that it's a slide. It will automatically look for a cover slip, which has been correctly identified. Plus, then another segmentation will run to identify the tissue sections, which are here highlighted in yellow. And there will be a final net, which is then just doing and running a quality check and give you an indication on whether your sample is clean and or might benefit from some further cleaning. As you can see, we have identified our brain sections here, and it might be that there are some positions which are not 100% correct, like this one here, which are two sections very close to each other. So of course, this is all interactive objects, so you can easily just make use of functions here like editing. So cut them away, get them, for example, separated here, or if you wish, you can also add manually further regions of interest. And of course, you can also add it in that way that, for example, and that's what I'm doing here for the reason to save some time, that you're just going with those three central sections over here. In the next step, we are directly jumping over into a second step of this wizard. And by the way, this wizard has also been improved here in the Zen 3.4 version with the direct link into the pre-scanning function. But before we go into the preview scan, we will run an automatic focus detection, and that is being based on the far red LEDs, which Gerd has introduced to you, which are being located in an additional position in our condenser. And now we're executing 3 long-range autofocuses, and basically always in the center of our section, running a focus approximation. And as you can see on the upper left side, get the individual Z value for each and every of the sections here, and which allows us directly to go into our next step, which is here, for example, to run a preview scan. And this is what I want to do in the next step. So we are on a 5x magnification, and another nice feature that we introduced, technically introduced with the AI sample finder, that's a contrast technique called coherence contrast. And this coherence contrast is based on the coherent illumination, the far red illumination in the condenser, and basically allowing us to give a very crisp contrast, and allowing us to do to run, for example, in an experiment or as a preview scan, this contrast image here. So let me just show you how this looks in live. So we are on a 5x lens and get some very nice contrast image here. And in here, we can also run like a shading correction, and then directly jump over into our preview scan step. As you see, now the preview scan started. And we're just running a preview scan based on this coherence contrast image with a 5x magnification on 3 of the sections here on the slide. And let me just zoom in a bit so you get an idea on how good the contrast image already is. And this could be already sufficient for us to assign further experimental regions based on morphology of the tissue sections here. And this would be the next starting point for a subsequent experiment. Of course, if you want to go directly with fluorescence information, you can also use fluorescence channels here in a preview scan. Once we finish this wizard, we can directly choose one of our predefined experiment setups or create a new one from this dialog. So very streamlined experiment setup within the AI sample finder wizard to link out directly to our subsequent experiment. And this is what I want to do next. Let's go maybe to just one of those brain sections, maybe to the one here in the center. And besides the information we have already, we would like to add some fluorescence information as well. So let me just go to fluorescence channels, which I've already set up. Of course, you can also use the smart setup way and use the information from the database to get your channel settings set up. And let's have a quick look on our sample here in our live view. So basically, the sections are stained with DAPI on the one hand side, and secondly, there's a sparse neuronal labeling. So you can see a few neurons here, one here in the center, which are expressing fluorescence information. And in the next step, we would like to run a preview scan, for example, based on higher magnification to get those neurons very nice. And therefore, I'm switching here directly in the software, my hardware, and I'm assigning an experiment setup. So in this way, we can directly use the navigation and tiles dialog here, which is using then the information from the AI sample finder and our preview scan, and allows us directly to assign scanning regions. For example, just by using this polygon contour tool over here. Of course, we could also use multiple position settings here, and that's just a matter of taste and a matter of experiment, what you would like to set up. And just to show you what I'm about to image here, so we want to see the neurons on the one hand side, and secondly, we want to see the nuclei. But as you can see, our neurons are very much expanding into the tissue, so they're very much expanding also into the Z range, meaning we see a lot of out of focus blur. So for this experiment type, therefore, I'm assigning a tile scan, and besides the tile scan, I also want to assign directly a cleanup of the images, which is also a new functionality in Zen 3.4, which is called deblurring. So we can directly use the online processing functionalities we have, which is called a module named direct processing. And this is what you can find here on your left-hand side tool. And within the direct processing, I'm assigning now a function called deblurring, and we'll run this directly on the fly, and we'll have the software opening the images automatically after finalization. Besides deblurring, of course, the direct processing module also allows you to use other processing functionalities. For example, if you have an apotome in use, you can do the apotome raw convert. You can use some deconvolution settings to be run online, as well as denoising and unsharp masking. By the way, if you are operating a Eriscan equipped microscope, you can also use the Eriscan processing function as a direct processing option. All right, as you can see, I've assigned a tile region, and I just briefly mentioned there is also a new feature within this software release, which is a automatic distribution of focus support points. Those are the yellow spots you can see here in the tile region. And basically on those positions which have been distributed here with some algorithm, the system will, upon starting the setup, the acquisition will run according to the strategy here, an automatic detection of the focus points, and then run the acquisition through it. So let me start this experiment and let's watch what the system is doing. So as you see, initially there's a software autofocus running at the respective focus support points here. So you can see the stage is moving to the yellow dots here in our tile region, executing the software autofocus, what you can see here indicated on the left-hand side. And now just running a quick tile scan, and I'm extrapolating all the different Z planes according to the support points which we have distributed. And this is this is on the one hand side very fast, and secondly, it's very precise in terms of focus map. And you can also watch on the right hand side your objective revolver moving, so basically adapting for each of the tiles automatically the Z value. All right, so here we go with our tile scan. And as promised, we directly get a processed image, which is then our deblurred image. Let me just put this side next to each other. And let me show you how this is looking like. All right, so here we go and have on the one hand side our perfectly focused single plane tile scan. And on the other side, you can see that we directly get our online processed images, our cleaned up images using the deblurring functionality to get rid of some of the out of focus blur of the images. And in the background, I've directly written all this information here into a what we called Correlated Workspace. So this is a file which has been which we can directly write on the fly, which all the acquisition we are executing on our microscope, all the image data is being automatically put into, and this according to its spatial location, which directly allows us to browse our project data, this project file according to the images that we have acquired, and make use of this tool, for example, to do some re-investigation of the sample. So after a very quick and straightforward alignment of our data, which is also supported by a wizard, we can directly come back to the positions that we want to drive to. And of course, it's a tool for correlative microscopy, so allowing us not only to bring samples back onto the same microscope and put all the data into a single project file, but also allows us to bring data to another light or electron microscope.
Thank you, Frank. That is pretty amazing. I think if I had this in lab, it would have saved me a lot of time when I was still doing research. Like many of my customers and our customers, we pack a bunch of tissue onto one slide. And then you spend a lot of time focusing and finding and cataloging what image went where. So especially I think Zen Connect is very handy to keep all the data straight. I think everybody is probably wondering, how does this work for multi-well bleeds?
All right, so in the next experiment, let me show you how this looks like on a 96 well plate. Okay, so let me quickly clean up here, and then let me just insert a 96 well plate into the microscope stage. All right, so now I've just added a 96 well plate holder into our microscope stage with a 96 well plate in it, which has a cover glass bottom and has some adherent cells on it. So it's U2 or S cells. Okay, so for the next experiment, let's directly go to the next experiment setup, and let's start another round of AI sample finder. All right, again, just start the AI sample finder and start the sample finding process. Now we will have another round of reflected light illumination and detection with the small camera, which is mounted next to the objective turret. And the first neural network is now identifying that in this case, we're not working with a slide. Therefore, it chooses to go with another strategy. And the strategy which is being executed now is just a tile scanning using this reflected light illumination and detection. So we are not executing here the composite dark field contrast, but we are rather going for acquiring within a tile scan, as you can see it here, the whole bottom of the plate. And within the next steps of segmentation, the artificial intelligence in the sample finder is identifying the containers in our well plate. And that is very handy, because when we are exiting now the AI sample finder wizard, we will directly be in a calibrated experiment. Also here we can make use of the long-range autofocus, which is then doing a global correction of our Z-plane based on 4 different focus support points. And this will be a very good basis for our subsequent experiments. And as you will see in a minute, the experiment that we are assigning now are all being assigned in the so-called carrier-based mode, meaning that the carrier information coming from the area sample finder will be used for the calibration of the microscope stage, and will also be used directly to assign the correct well ID to our experiment data. Okay, as you see in our navigation and tiles overview, we do have now our 96 well plate with the identified carrier IDs, plus we have a graphical representation of our carrier, which we can then directly use to assign our experiments. So for example, what we could do, I've just switched here to a 10x magnification. We could go with a 2-channel fluorescence acquisition. And as you see, on the one hand side, we do have, again, a DAPI staining, so a nuclear staining of our cells. And secondly, we do have some of the cells in this plate, which have been transfected with a plasmid containing TD tomato. So doing here a labeling of actin using LiveAct TD tomato fusion protein, and this will be our second channel for the acquisition. So to assign our experiment, as mentioned, we can directly use this graphical representation down here, and then go into this carrier-based acquisition mode. And in this mode, for example, we can supply now either tile images, tile scans to the respective wells, or we can make use of positions or position arrays to be distributed either in like a center or a default way, or can also make use of the random function here. So let's just go for a single position of each well here. And last but not least, we want to have a focus strategy. So again, we want to make use of the autofocus. By the way, you can also make use, and that's would make a lot of sense, for example, in a time series experiment, you can make use of a hardware-based focusing strategy. So using our reflection-based and definite focus tool to directly run a very quick focus stabilization, for example, to keep the relative focus over time constant. All right, so that's the experiment we want to execute now. Let me just start this experiment, and now we can watch the system working. So we are moving here to the respective well, executing an autofocus based on our DAPI signals here. So as you can see, we're seeing here cells and nuclei in different concentrations. So pipetted this in that way that we have a different levels of concentrations and also expression ranges. And basically, you can imagine that if you would run that on the whole plate, that's just a single click to start the whole experiment, and the system has the global focus correction, which you can see here also with these white dots indicated. So those are the support points being picked to correct, like for a tilted plane here in our 96 well plate. And now we are already done with our acquisition. So meaning the AI sample finder really helps to set up experiments like that, because the calibration of such a carrier is directly being done by the AI Sample Finder in the initial Sample Finder process.
Thanks, Frank. That was incredibly quick and easy. I actually see that there's a question in the chat that seems relevant to what you just showed, so I'm going to go ahead and ask it now. Can Zen easily do cell counts in each well?
Yeah, yes, we can do that. Basically, as you might know, Zen always had a image analysis functionality, which is also quite sophisticated, if you like. So meaning we can also include machine learning and deep learning functionalities. But there is, and we added this with this recent version of Zen, single click predefined image analysis settings. For example, we can directly run an analysis here, which is basically using 2 different segmentation steps, and we can directly run a cell count, for example. This is one thing that you can directly do, or as what I started here now, we can directly do a analysis of a gene and protein expression. And this would make a lot of sense in this type of cells, because here we had a transfection regime with different concentrations, and we would be interested now to maybe get the readout here as a graphical representation of our transfection efficiency. So if we have titrated our transfection reagent and would like to know which of the treatments would be our best setting to go for in our next experiments, we could directly extract from the seed map that in E6 we had obviously the best transfection, so meaning the highest rate of positive cells among the total cells, which here was around about 30%. So we have those single click analysis features, and basically they are all built up on the internal Zen image analysis machine, also allowing you to use high-end algorithms and make use, for example, of artificial intelligence and make use of some pre-trained neural networks.
Thanks, Frank. So the bio apps, they look really powerful and easy to use. I love streamlining what would ordinarily be a complex image analysis, including artificial intelligence, into a simple, easy to use app. It looked like you just literally clicked one button and then you got this great analysis, including a heat map display very quickly. So again, thanks for showing us both those examples. Looks like we have some time, a few minutes left to take some questions. Please share your questions in the questions function if you haven't done so already. So I think is Gert still there with you? Can I address questions to both of you? Okay, great. All right, so let's start with this one. I think since Gert did a nice job explaining what is needed to add AI Sample Finder to one's inverted microscope system. So there's a question here. Can these features, and I assume we're referring to everything Frank showed, the Sample Finder, Zen Connect, be used on confocals as well as the wide field system?
Good point. The answer is yes. If your confocal comes with an Axio Observer 7, as you already stressed out, if it is an LSM 900 or the latest 980, your confocal can benefit from AI Sample Finder as well from Zen Connect and the bio apps we quickly had on display. And you can retrofit existing confocals as well. So if it's a 900, if it's a 980 on an inverted stand, why not?
How about? Oh, okay. Thank you, Gert. How about Zen Connect? Is that something that's available on all platforms?
yeah, it's available on SandBlue again. And you can do something like Frank already has shown quite nicely. So you can start a new project, run your experiment, do precisions, whatever you like to do. You can zoom in and out, and everything will get handled at the end in a single file. So all the data are still in context and nothing is going amiss. This, if you like, somehow replaces your logbook. So you don't need to be aware all the time what you did and what sample and what the naming was and everything. It's one file. But your question, so it's quite clear on one system, right? But you can do much more than this. So assume you only have one system equipped with the SandBlue software, and you do some super resolve, whatever imaging. You can easily load into that software platform images from wherever. So maybe a snapshot by your smartphone. It can be an image out of an electron microscope. It can be one from a dissection scope. If the other system comes equipped with a scanning stage, you can do some auto alignment. If not, you do have all the tools handy to do the alignment and make images transparent and do some overlays and everything like this.
Yeah, that's great. I think Colleen alluded to the fact that she would have loved to have something like this when she was still in the lab, just for documentation purposes and knowing what tissue you imaged before on a slide that has 12 sections on it. So this is great. There's a question here. I believe they're basically referring to the AI sample finder. The new equipment. They asked if you could elaborate on the optics and lighting schematics. I assume they're referring to the 32 or the 128 LED array. So maybe, Gert, since you also went over that in your presentation, if you can maybe elaborate a little more on that.
Yeah, so we have this 128 LEDs due to some patent reasons. It could have been one more, give or take. Yeah. So we do them one by one. So quick you don't see it, taking 10 seconds or so, and you get this overlay of 128 images as this composite dark field image at the end. It's already composed once you're done with this, right? After a couple seconds, it's eliminating a sample from very different points of view, never in the direct way. Okay, and this makes things available like this translucent samples, which you will never see in any kind of transmitted light or reflected light contrast. So dark field helps a lot. And I guess the basic idea behind this was to give us access to any kind of sample, whether staining or no staining.
Yeah. Great, thank you. Oh, go ahead.
Second, this is what Frank pointed out at the end, is this LEDs, which are somehow based in the beam path of the condenser, used for focusing. These two guys can, or at least it's more than two, can be used for giving this very unique contrast we have seen at the very end of this overview image, which nearly looks like a DIC image or something. Yeah.
So you're referring to the coherence contrast, and that's part of the whole AI sample finder condenser apparatus has, both the LED and the IR light source. So it is a really unique solution from Zeiss. It is a special condenser that goes on our Axial Observer 7, and it's actually quite reasonable in cost. It just imparts so much, functionality and ease of use to the system, and we often ask why not? Why wouldn't you why wouldn't you get this if you had an inverted microscope? So so thanks for answering that question. So maybe a question for Frank, since he did such a nice job showing the bio app. There's a question: are there other bio apps available, and what can they do?
Yeah. So far we do have 4 different bio apps, and they are covering like most of the standard measurements you would like to do directly on your sample. So it's cell counting, it's a confluency measurement. So cell counting is being based on fluorescence information. You have the option to go with a pre-trained neural network or use some algorithm for thresholding. The confluency bio app is being set up for contrast images. We have the gene and protein expression bio app. This is the one you have seen, where we have two different layers of segmentation. So the requirement for that would be basically 2 channels to run an analysis. In the background, it's based on a analysis feature which we have introduced some 2 years ago, which is called zone of influence. This is what you see here around our nuclei, the red rings. And there is a 4th bio app available, which is called spot detection. And basically the spot detection bio app is another bio app where you have 2 different layers of segmentation. So again, the first one is segmenting a primary object like nuclei, either based on a neural network or on an algorithm-based thresholding. And secondly, identifying spots within this object. And this is, for example, an assay where you would like to go with foci within the nuclei, do some foci counting, like single strand break, DNA strand breaks, etc. And this is where we have set up the spot detection bio app for.
Great. Go ahead.
Yeah, sorry, Sam. Just to mention, as mentioned, they're all based on our standard image analysis wizard. So you can directly go from your bio app analysis back into the analysis wizard. For those of you who would like to maybe add another layer of objects or classes, you can directly use the bio app information and get started with a new experiment image analysis setup.
And are there plans to add any more bio apps in the future?
Yeah, of course. This would be very nice, and this is very much depending on the market requirements, I would say. So therefore, please test the bio apps, use the bio apps, feedback to us on what is needed and how to continue. But as you see, the framework is there, and implementation of more bio apps, of course, would be more than welcome from our side.
Excellent. Yeah, I'm really excited about these new bio apps. And as you said, you know, as our customers start using them, we would love to get your feedback and some input as to what else do you need. You know, what other analysis workflows would be helpful to have in this easy, almost just single click app to generate your, you know, basically to go quickly from your data to results. So. Okay, so there's a question here about the deblurring processing you showed. I don't know if it would be possible to bring that image back up or not. I saw that you had closed it.
Mm-hmm.
So while you're doing that, I'll go ahead and ask the question, which is, I'm curious to learn more about the deblurring processing you showed. Can you tell me more how this is processed or calculated?
Yeah, sure. So what the deblurring is doing is basically calculating 2 different planes, 2 additional planes. So the deblurring is working on Z-stacks as well as on single plane acquisitions. And what the algorithm is doing, and we are very clear in that to really tell you what the algorithm is doing in the background, it's calculating 2 different additional planes which are being convolved from the original image. So basically we are calculating the blur information from our focus in image. And those two images, those convolved images, are being subtracted from our original image using a nearest neighbor algorithm. So it's a very straightforward algorithm. And by the way, we provide this deblurring algorithm already within our Zen Lite version. So with our free version of Zen, you can directly use this deblurring functionality. The only thing, if you would like to run this online as we did, you need the framework of the module direct processing, which you can set up on the same machine, which you can set up on a network machine, And this is an additional module, which is not in the free version, but for an offline like processing of your data from your microscope, we provide this algorithm already within the free of charge version of Zen. And so here is the image that we generated in the experiment here. Just did a quick stitching of our tile scan. This is what I was running here in the background on the left side. Let me just show this here in this split mode view. And as you can see, we removed a lot of background information in this image. We took a single plane. Of course, those processes are very much going into the tissue. But if you compare that over our initial wide field image, and this is what I'm showing here, you see that first of all, we have a much higher background, and overall much higher background. And secondly, also much more of blur information into our image. And this is what we can easily remove to some extent using this online processing function, or also as an offline processing. Of course, it's not a pure optical section, but if you, for example, would like to upgrade your wide field setup with the deblurring option, which is a very fast option, you can also do true optical sections using our Apotome, which is a hardware device generating a structured illumination and allowing to calculate true optical sections.
Thanks, Frank. I didn't realize that the deblurring was available in the ZenLight software. Just so everyone knows, I put in the chat the link to download ZenLight in case you're interested in trying out that functionality. So that's very cool. I didn't know that, so I learned something today too. We're going to wrap up here soon. There was maybe one or two questions actually that about whether you can combine AI sample finder with live cell incubation. So maybe you can just give a quick answer to that question.
Yeah, so the answer is yes, you can do that. Because as you see, the AI sample finder, that's designed in the way that you can easily put an incubator around it. So all the additional hardware components like the camera, the LED illumination sources, they do not interfere with any of your incubation solution. May it be like a large incubator, the XXL incubator setup, or even the small incubator, the incubator S for the stage, so the stage insert incubator. All of that works with the AI sample finder illumination and detection.
Okay, great. I think we actually got through all of the questions. Yeah, let's just give Gert and Frank a big thank you. So Gert, thank you for the great, presentation, and Frank for the excellent demonstration of how easily and how quickly one can go from literally placing their sample on the microscope to quantitative results. I was especially impressed with the well plate example. Setting up a quick screening of the wells followed by calculating transfection efficiency and then displaying it as a heat map. I think you did that in all under 5 minutes. This is really powerful, and it's so cool that this can be done in both wide field and confocal systems on the inverted platform. Like I said, I think we addressed all the questions. I'll go through them again. If there's anything I missed, our team will reach out to you directly and provide answers and discuss your needs. If you're interested in getting a demonstration of these features with your samples, please reach out to your local Zeiss account manager or specialist, or email info.microscopy.us@zeiss.com. I'll put this in the chat here in a second, so you'll have that as well. But otherwise, again, a big thank you to Gert and Frank. Thank you all for your attention and attending. Let me just put this in the chat before we end the webinar. But I just want to say thank you, everybody. Goodbye. Stay safe, stay healthy, and have a great rest of your day.
Thank you, everyone.
Thank you.
Thank you.